So I want to thank everyone for joining us today. I'm stepping in for Joe as the moderator. It gives me a great pleasure to introduce Niamh Mullins, who is assistant professor in the Department of Psych and also in the Department of Genetics, and she's a key recruit into the Mount Sinai Clinical Intelligence Center. Niamh's lab, for those of you who don't know, is really now probably the biggest key driver of our large scale genomics efforts in psychiatry here at Mount Sinai. She is the lead. She has been the lead data scientist for the PGC bipolar working group, which is the largest genetic study of bipolar disorder in the world. That she is also a founder of the the International Suicide Genetics Consortium, which is again like the largest study of the genetics of suicide in the world. So she's really just a key figure in the field. She got her training at the best places, PhD at King's College, postdoc here at Mount Sinai in the Pamela and Eli Stahl kind of a family of scientists. And really it's just we're just thrilled that she decided to stay on to build her lab here at Mount Sinai. And as you'll see today, she's a real star in the field. So take it away, Niamh.
Thank you so much, Alex, for that very generous introduction. And I'd like to thank Joseph Buxbaum for inviting me to give this talk today. So this is a great opportunity for me to introduce some of the work that my lab is doing. And I realized that this is my first talk as a faculty member, and I'm still getting to know more of you in the psychiatry department, as well as some folk in genetics as well. So I thought I'd start out by giving a brief overview of my lab generally and the work that we are doing. So these are the core research themes in my lab. As Alex mentioned, I work on bipolar disorder genomics in the Psychiatric Genomics Consortium. We led a large GWAS last year of bipolar disorder, and since then we have been working to increase this further in scale through collaboration with 23andMe, as well as having a major focus on increasing ancestral diversity in our studies. So in addition to this, we've gotten very good at identifying genetic risk loci for psychiatric disorders. But one of the key challenges is to pinpoint the likely causal genes and variants within these windows of association. And so that's why my lab is particularly interested in fine mapping genetic risk loci using statistical and functional fine mapping methods and integrating omics data. And we have a project underway at the moment focusing on fine mapping bipolar disorder risk loci. One of the other major themes is suicide genomics, and that's what I'll be discussing today. This involves doing large-scale GWAS and bioinformatics follow-up of suicide phenotypes in the ISGC and the PGC. And we're also working on developing clinical and genetic risk prediction models for suicide phenotypes in patient populations using the EHR at Mount Sinai, as well as other healthcare systems in the US. And as with most of this statistical genetics work, we need huge sample sizes to do this. So we collaborate widely internationally on these projects. So in addition to these core projects in the lab, I also have a few other collaborations that I'm involved in at Sinai. These are really driven by other labs, but provide some great opportunities to team up with colleagues here and to work on some novel projects. So this is an outline of the talk today, which will focus on the suicide genomics theme. I'll start by giving you some background on genetic epidemiology to tell you why I'm working on this project. I'll present a GWAS of suicide attempt in the International Suicide Genetics Consortium and a meta-analysis we are working on with Million Veteran Program. I'll briefly touch on the genetic relationships between these suicide phenotypes, and finally I'll introduce the new suicide working group of the PGC and tell you a bit about our future plans. So as I'm sure you know, suicide is a huge public health problem. It's responsible for over 700,000 deaths per year worldwide. And it's estimated that in the US in 2020, there were approximately 130 deaths per day due to suicide. It's a leading cause of death overall, but particularly amongst young people. It's the second leading cause of death for individuals between 20 to 34 years old. But it's still not exclusively a problem of the youth. Over a third of people who died by suicide in 2020 in this country were over 55 years old. And it's estimated that for every death by suicide, there are up to 25 times the number of non-fatal suicide attempts. And suicidal ideation is even more common. It affects over 9% of the population. Over 90% of people who die by suicide have a diagnosable mental health condition at the time of their death. But of course, most people who have a mental health condition never make a suicide attempt. And it's estimated that suicide costs the US economy about $70 billion per year in healthcare and work loss related costs. And of course, on top of this, there's the huge societal burden of suicide to patients, families, and communities. And for all of the statistics here, the true rates are actually likely to be higher given the underreporting of suicide due to the stigma that still surrounds it. In terms of treatment, it's now quite well accepted that suicide-specific treatments and interventions are necessary, as well as treating a primary psychiatric disorder. And most relevant to my research in genetics would be the use of medications as treatments. And there's only one FDA-approved medication for treating suicidal behavior, and that's clozapine, but it's only indicated in schizophrenia. Lithium has been reported for decades to have anti-suicidal effects in mood disorders. And more recently, there's a lot of interest in using ketamine and the nasal antidepressant esketamine as treatments, with growing evidence that they reduce suicidal ideation independent of their effects on mood symptoms. There's also some evidence for the use of SSRI antidepressants, although they may have some age-specific effects. So while there's some medication options, there's many limitations on who these medications can be prescribed to. There's a lack of large RCTs demonstrating effects for some of them. And of course, they have many side effects as well. So there's an urgent need to develop new medications that are rapid, safe, and effective for treating suicidality in the diverse group of affected individuals. So before I get into the genetics, I just want to take a minute to clarify some of the phenotype definitions that I'm using, because there's a lot of closely related but distinct phenotypes here. So suicide is intentionally ending one's own life. Suicidal behavior is defined as behaviors that may result in ending one's own life, whether fatal or not. And this includes suicide attempt as well as preparatory acts, but it excludes suicidal ideation since thoughts are not a behavior. Suicide attempt is defined as a deliberate act of self-harm where there's at least some intent to result in death, although it's not fatal. And that suicidal intent is a very important part of that phenotype, and it distinguishes suicide attempt from non-suicidal self-injury. Suicidal ideation is thoughts about ending one's own life or wishing to die. And collectively, these four phenotypes can be referred to as suicidality. So that's a broad term encompassing all of them, but it's non-specific. And then two related phenotypes are self-harm, which are self-injurious behaviors where there may or may not be intent to die. It does not distinguish between those two. And non-suicidal self-injury, where there are self-injurious behaviors, but without intent to die. And the intent is to relieve negative emotions. So as you can see, the phenotype definitions can be quite complex. Studies investigate a variety of phenotypes, which can make it somewhat difficult to combine information across studies at times. And while these are symptoms of psychiatric disorders included in the diagnostic criteria for MDD, for example, in the DSM-5, suicidal behavior disorder was introduced as a condition in itself for further study. And so in terms of what we know about the genetic etiology, the twin heritability of suicidality, so that broad phenotype, is 30 to 55%. And this is from a meta-analysis of 32 twin studies. In the figure on the right here, I'm showing the twin heritability estimates for a range of psychiatric disorders for comparison. So for disorders like bipolar disorder, autism, schizophrenia, heritability is up around 80%, whereas for disorders like major depressive disorder and anxiety, it's about 30 to 40%. So the heritability of suicidality is much more similar to that of MDD or anxiety. But these twin studies very consistently indicate that this heritability is not explained by psychiatric disorders alone. And one twin study which attempted to adjust for psychiatric disorders estimated heritability of suicidality in the range of 17 to 35% after controlling for psychiatric disorders. In terms of having specific heritability estimates for suicidal ideation, attempt, and suicide, estimates in males or females separately, and estimates across the lifespan, we don't have reliable estimates for these from twin studies because of the low base rates of these phenotypes, which mean that the data are quite sparse. So this is actually an area where we need to use molecular genetic data to tackle these questions. And while my talk is on genetics, I want to emphasize that environmental risk factors also play an extremely important role. So since these genetic epidemiology studies established the genetic etiology of suicide attempt or suicidality that's partially distinct from that of psychiatric disorders, many genetic association studies have been conducted to identify the specific genes and genetic variants involved. There have been, of course, many candidate gene studies over the years, but I will start from the GWAS era. These are some of the studies that have been published over the years on suicide attempt, suicidal ideation, or suicide, and a smaller number looking at severity of suicide attempt or suicidality as an ordinal phenotype. And the classic way that these studies have been done is to take patients ascertained for having mood disorders and split them into those with a history of suicide attempt versus those without a history of suicide attempt, and use this sort of within case design to disentangle the genetics of suicide attempt from that of the mood disorders themselves. And this table shows the growth of these studies over the years. Up until 2020, the sample sizes were still quite modest. And so there were some genome-wide significant loci from these studies, although a lack of replication across them. However, GWAS did find small but significant SNP heritability estimates for suicide attempt in the range of 4%, and one estimate for suicide was 16%, so considerably higher SNP heritability. Another finding from these GWAS was polygenic overlap between suicide attempt and, in particular, major depressive disorder, with several studies reporting genetic correlation between suicide attempt and MDD between 0.5 to 0.8. And in this figure here, what I'm showing is that polygenic risk scores for MDD are significantly higher in patients with a history of suicide attempt versus patients without a history of suicide attempt. And that's true within MDD, within bipolar disorder, and in schizophrenia. So these studies have confirm the evidence that the genetic etiology of suicidality is partially shared and partially distinct from the related psychiatric disorders. And this is something that we have considered very carefully in the design of newer and larger GWAS of these phenotypes. So in 2018, during my time as a postdoc here at Mount Sinai, I co-founded the International Suicide Genetics Consortium, along with my colleague Doug Rudifer, who is based at Vanderbilt. And we recognized the real need for a consortium-based approach to do these genetic association studies and really increase sample size and accelerate discovery. And we decided to focus our initial study on a GWAS of suicide attempt. We had huge enthusiasm from colleagues in the field to come together and contribute to this effort. And these are all of the cohorts included in our first GWAS. So it was really a worldwide effort. We had 18 cohorts represented. And on a practical note, how we did this was by having analyses conducted at each site by the local research teams and having summary stats shared with the ISGC. We developed standardized case and control definitions and analysis protocols, which we distributed across the sites to ensure rigor and comparability across them. And we did within ancestry GWAS, followed by multi-ancestry meta-analysis across the sites. And we had a big focus on promoting ancestral diversity in this working group from the outset. So across these 18 cohorts, we had almost 30,000 cases of suicide attempt and over 500,000 controls. In terms of the case and control definitions, 80% of cases in the study are of suicide attempt, so that's a non-fatal attempt. And this information was typically obtained from structured psychiatric interviews. And we had 2 studies that used self-report and 3 that used hospital records. But regardless of the method of assessment, evidence of suicidal intent was a very important part of the phenotype definition. So we're not looking at self-harm here or non-suicidal self-injury. 20% of the cases in the study are individuals who died by suicide, so we decided to combine those 2 phenotypes in this meta-analysis, and these cohorts were collected through collaboration with coroners' offices. The cases in the study are mostly individuals who were ascertained for studies of psychiatric disorders, and they also have information on lifetime suicide attempts available through their structured psychiatric interviews. Although we have a handful of studies that were actually collected for research on suicide attempt or suicide, as well as some population biobanks. And then across the studies, controls can be best described as a general population sample. So they're screened for the absence of suicide attempt if possible, but they're not screened for the absence of psychiatric disorders. So this schematic here on the right should illustrate the case control comparison that we're doing. We have cases of suicide attempt, the majority of whom have a psychiatric disorder, versus controls, some of whom have a psychiatric disorder. And so there is a big difference in the prevalence of psychiatric disorders between those 2 groups, which could potentially confound the GWAS. So we later used a statistical conditioning method to be able to adjust for this. And so in the GWAS, we found 2 genome-wide significant loci, one in the MHC and one in an intergenic region on chromosome 7. The SNP heritability of suicide attempt was 6.8% on the liability scale. So then in order to adjust for that difference in the prevalence of psychiatric disorders between the cases and controls, we conditioned the GWAS results for suicide attempt on major depressive disorder using GWAS summary stats from the PGC and the MT-COJO method. So the results after conditioning can be interpreted as SNP effect sizes on suicide attempt not mediated by MDD. And this is the picture after conditioning suicide attempt on MDD. We saw SNP heritability roughly halve, so indicating the genetic etiology specific to suicide attempt remaining after conditioning. The association in the MHC disappeared, but the chromosome 7 locus remained genome-wide significant. And when we also conditioned on other psychiatric disorders in addition to MDD, there was no further change to these results. So very briefly, we also implemented a GWAS of suicide attempt within psychiatric diagnoses. So that within case study design that I mentioned has typically been used in the past. We were able to do this across 13 cohorts in the ISGC. We had a much smaller sample size here because you need a more specific type of cohort to implement this study design. But we found from this a SNP heritability estimate for suicide attempt, which was on par with our estimate after we had conditioned on MDD. And we also found total genetic correlation of this GWAS with our GWAS of suicide attempt conditioned on MDD. So this validates our statistical conditioning approach, and it illustrates that we can achieve the same result as this within case study design using statistical conditioning, which is a much more flexible approach. And this is a closer look at that chromosome 7 intergenic locus. So first, we directly replicated this association in a cohort from Million Veteran Program of over 14,000 cases of suicide attempt, making this the first genome-wide significant locus for suicide attempt to actually replicate. In the forest plot here, I'm showing the effect size of the top lead SNP at the locus on suicide attempt and other psychiatric disorders or traits. So it has not been associated with any psychiatric disorders previously from the largest GWAS done by the PGC. But interestingly, it is a genome-wide significant hit for insomnia, smoking, and risk-taking behavior. It's in an intergenic region on chromosome 7. We have been working on dissecting this region for some time now using various approaches to try and pinpoint the likely relevant gene in the area. So we've been using approaches like integrating QTL data, chromatin loops, some whole genome sequence data, some post-mortem gene expression data. We've seen some preliminary evidence pointing towards the FOXP2 gene, which is upstream. But while the association is statistically robust, at the moment we've not clearly linked the variants to any of the nearby genes. So this effort is ongoing at the moment. Here I'm showing the genetic correlation of suicide attempts with a range of psychiatric traits or disorders before and after conditioning suicide attempt on MDD. So there's widespread genetic correlations between suicide attempt and these psychiatric disorders, especially with self-harm ideation, MDD, PTSD, and alcohol dependence. And after we had conditioned suicide attempt on MDD, these tended to decrease, but they still remain substantial. And I just wanted to highlight that even after we conditioned suicide attempt on MDD, it remains genetically correlated with MDD. And that might seem a bit counterintuitive at first, but this genetic correlation remaining represents pleiotropy, so genetic variants that have effects on suicide attempt and on MDD. And we know there's pleiotropy between all of these psychiatric disorders, so we expect there to be pleiotropy with suicide attempt as well. Here we looked at genetic correlation with several hundred other risk factors or medical disorders via LD Hub. We saw positive genetic correlations between suicide attempt on smoking, pain, risk-taking behavior, sleep disturbances, many measures of poor overall general health from the UK Biobank. And this figure here shows the top 30 genetic correlations with suicide attempt. And most of the top correlations were psychiatric traits. And after we conditioned suicide attempt on MDD, these tended to decrease. However, but for non-psychiatric traits, the genetic correlations were largely unchanged by conditioning on MDD. So this suggests pleiotropy between suicide attempt and these non-psychiatric risk factors, which is not mediated by depression. So we published this paper last year in Biological Psychiatry, We had over 250 co-authors here. It was a huge team effort. I'd like to acknowledge Jay Kang, who is a graduate student in Doug's lab, who was co-first author of this with me. Of course, Doug, we set up the ISGC together, and our GWAS summary statistics from the paper are available online. We have a short application form. We felt that given this is such a sensitive topic, that we had a responsibility to ensure appropriate and ethical use of the data and have a safeguard against any potential use that could cause harm. But we have approved all of the applications to date, which have all been for research purposes. So moving into the next section of the talk, while we were working on this study in the ISGC, we became connected with some colleagues at Duke University who had a similar effort underway in Million Veteran Programme. And MVP is a biobank of over 800,000 veterans from across the US with DNA and EHR data available. And our colleagues had conducted a GWAS of 14,000 cases of suicide attempt within MVP. This Manhattan plot is from their multi-ancestry GWAS, where they found 2 genome-wide significant loci. MVP is quite an ancestrally diverse cohort, so they also had some other GWAS hits in specific ancestry groups, some of which replicated in the ISGC data. So we've had a very productive collaboration with these colleagues for the last year or two. We've been able to do reciprocal replications for each other. And given the large sample sizes that we had both achieved, the logical next step was to do a meta-analysis between the ISGC and MVP to further increase our power. And this slide shows some of the key characteristics of the two studies. They're comparable in many ways, but MVP, as I said, is quite ancestrally diverse. It had a substantial proportion of individuals of admixed African American ancestry, as well as Latino ancestry. And the major difference between the two cohorts, of course, is that MVP is a veteran-based cohort, whereas ISGC is largely civilian. We only had one veteran-based cohort included, which was Army Stars. So there are major differences in environmental exposures between our cohorts, as well as gender differences, with MVP being predominantly male and ISGC having higher proportion of females, as we typically see with these kind of genetic studies. So in terms of doing a meta-analysis between our cohorts, one of the first things we were interested in was the genetic correlation of suicide attempts between the ISGC and MVP as a whole. And we found this was not significantly different from one, which indicated the shared genetic architecture between the data sets and suggested that our meta-analysis is unlikely to be substantially impacted by heterogeneity in phenotype definitions or ascertainment differences. So we conducted a multi-ancestry meta-analysis, including almost 44,000 cases of suicide attempts. And we're now starting to have sufficient numbers to be able to do ancestry-specific meta-analyses as well. So in the multi-ancestry meta-analysis, we have found 8 genome-wide significant loci. The most significant here was, again, that intergenic region on chromosome 7 from the ISGC1 study, which became even more significant here in the meta-analysis. We've also found several loci here that have been associated with psychiatric disorders previously. For example, the DRD2 locus, the furin locus, although these labels here refer to the nearest gene to the index SNP. So we have work ongoing to do detailed dissections of each of these loci to investigate the likely causal gene or variant and disentangle that from the genetic associations with psychiatric disorders. But there was no significant heterogeneity across the cohorts for any of the GWAS hits, and we found a SNP heritability estimate of 5.7% for suicide attempt. In the European-only subset, again, we found 8 loci, 4 of which overlapped with the multi-ancestry meta, and 4 additional loci. And we saw a slightly higher SNP heritability estimate of around 7%. In the admixed African American and the East Asian meta-analysis, there were no GWAS hits here. The sample sizes are much smaller, but still we found significant SNP heritability estimates in both ancestry groups. And I'm not showing the Latino ancestry here because that was only from MVP, and those data are already included in their published paper. So with these new data, we can now start to address questions about the biology of suicide attempt that we were not powered to look at in ISGC1. So one, for example, being which are the relevant tissues involved in suicide attempts. And here on the left is the results from a magma tissue set enrichment analysis, where we found enrichment of suicide attempt association signal in the sets of genes expressed in all of the brain tissues from GTEx and no other tissues. So this is a similar pattern to other psychiatric disorders where we see enrichment for all of the brain regions. And determining the relevant cell types remains a future direction for us. We're slightly underpowered to do that analysis at the moment. On the right is the integration of our GWAS results with EQTL data from brain tissue using the SMR method. So here we've taken EQTL data from the MetaBrain Consortium, which is a large meta-analysis of gene expression across brain tissues with almost 3,000 European samples. And we found one significant gene here. So what this result is suggesting is that alleles that increase risk of suicide attempt act through increasing expression of the FES gene in the cortex. And FES was within one of the genome-wide significant loci on chromosome 15. It is within the furin locus that I mentioned. It is encoding a protein with tyrosine kinase activity, so it's quite a broad function, but it has been suggested as a therapeutic target many times for some forms of cancer. It has a role in cellular differentiation. So I expect that we will see many more genes acting through this sort of mechanism as the power of our discovery GWAS increases. A topic of major interest to us is to look at drug targets for suicidality. So here we're looking at enrichment in genes targeted by different classes of medications, which we have defined using their ATC codes. And the gene targets of the medications are obtained from databases like Chembl or the drug gene interaction database. So this is basically like doing a pathway analysis, but instead of genes in the pathway, we have genes targeted by these classes of medications. So here we see enrichment of suicide attempt association signal in genes targeted by antipsychotics, which also includes mood stabilizers within that class. We also see enrichment in the targets of psychoanaleptics, its subclass antidepressants, and its further subclass other antidepressants, which if you look up the ATC codes are those that are not SSRIs or not MAOIs. And we can also do this analysis looking at the targets of individual medications. We examined over 800 of those. We didn't see enrichment for any individual medication. But again, I expect that as the power of our GWAS increases, we will find additional medications with significant enrichment and see this pattern of enrichment refine. And one thing to keep in mind here with this kind of analysis is that there's no direction of effect from this. So it cannot give us information on whether the effects of these medications would be to improve or worsen suicide attempts. And we have to use other lines of evidence to investigate that. But doing this sort of work could point towards potential opportunities for repurposing some classes of medication for treating suicidality. And this slide shows the genetic correlations with suicide attempt from this GWAS and a range of psychiatric disorders or risk factors of interest. So again, we see positive genetic correlations with all of the psychiatric disorders, most notably with major depression. We also see positive genetic correlation with chronic pain and insomnia, which are risk factors for suicide attempts. So these results suggest some shared underlying biology between suicide attempt and these disorders. We're also very interested in developing genetic risk predictors for suicide attempt that could be combined with clinical risk predictors in patient populations. And so here I'm showing the performance of PRS trained based on this GWAS in 6 target cohorts. So we pull out each of the target cohorts one at a time to make sure we have independent training and testing sets. At the moment, PRS for suicide attempt explain about 1% of variance in case control status in European ancestry cohorts. So this would be on par with some of the earlier GWAS of, say, schizophrenia or major depression before sample sizes were as large as they are today. But you can see that the performance is much poorer in non-European ancestry groups, and this really highlights the need to continue to increase ancestral diversity in our discovery GWAS to ensure that any future genetic risk predictors developed are broadly applicable across populations. So this is a summary of our suicide attempt GWAS to date. We have seen total genetic correlation of suicide attempt between civilians and veterans in this study, which was really encouraging to us for future planned work to further increase sample size and our ability to combine data from diverse types of cohorts. We see evidence for involvement of all of the brain regions, as well as enrichment in genes targeted by antipsychotics and antidepressants. I wanted to just briefly touch on some of the other GWAS out there of suicidal ideation specifically and suicide. So here I'm showing the recent results of a GWAS of SI in MVP. And this is unpublished work that has been shared with me by our colleagues. So they've done a GWAS of almost 100,000 cases of suicidal ideation within MVP, found 5 GWAS hits and a small but significant SNP heritability estimate for suicidal ideation. Here is also the Manhattan plot from the largest GWAS of suicide, which has been published to date by our colleagues at the University of Utah, which found a couple of genome-wide significant loci and showed that higher SNP heritability estimate for suicide. And this sample has been collected at the University of Utah through a decade-long collaboration that they have had with the coroner's office, and it continues to increase in size with a projected increase to 10,000 cases within the next couple of years. In terms of what we know at the moment about the genetic relationships between these phenotypes, using genetic correlation analysis, we have seen a strong but incomplete genetic correlation between the 3 of them. There is no overlap of the GWAS hits yet, but that would be expected given the total number of hits given our current sample sizes. But these results indicate some differences in the genetic etiology of these phenotypes and therefore likely the biology underlying them, rather than one of them simply being a more severe manifestation of another. And so we have further plans to dissect the genetic relationships between them, as this could uncover risk factors that could lead from transition from one of these phenotypes to the next. So for the last part of the talk, I'd like to tell you a bit about the new suicide working group of the PGC and the future analysis that we have planned. So I'm sure many of you are familiar with the PGC. This consortium was established in 2007 to unite researchers working on psychiatric genetics and really accelerate discovery in this area. The PGC has work groups for 11 psychiatric disorders, has published hundreds of papers over the years, including many of the landmark studies in our field, such as GWAS of schizophrenia, for example. But the PGC has not had a focus on suicide research. And so given the clinical importance of suicide and also the specific considerations with analyzing these phenotypes, dissecting their genetics from that of psychiatric disorders, the ISGC was recently invited to become the suicide working group of the PGC. And so I'm co-chairing this new working group along with Doug and another colleague, Anna Doherty, at the University of Utah. And our goals remain the same: to uncover the genetic and biological etiology of suicidality. And now through the PGC, we have a lot of new avenues of outreach, leveraging their global presence and leveraging their infrastructure to further scale this up and expand the scope of our work to achieve these goals. So these are some data from a recent grant submission that we did to fund this working group. Within the next 5 years, we are planning to do GWAS of suicidal ideation, suicide attempt, and suicide. And these are the projected numbers of cases in the study, including substantial proportions of non-European ancestries. 21 of these cohorts are new since ISGC1. And I would say they're all very tangible numbers that have been provided to us with sample descriptions by collaborators. So I think that with our continued outreach through the PGC, it's possible we'll even exceed these numbers within the next few years. We've also been working on developing a catalogue of biospecimen sequence data and omics data from these cohorts so that we will be ready to use that for targeted follow-up of findings from our main study. And one of the great things about having such a diversity of cohorts included is the variety of types of data that they have to do targeted follow-up, as well as facilitating collaborations between members of the consortium. So in our planned work in the immediate future, we'll be able to generate these large GWAS resources for suicide phenotypes for future research and for use by the psychiatric genetics research community. The goal here is not just to increase sample size and find more GWAS loci. These are really a resource. It's a means to an end. We want to use these results to understand the genetic relationship between suicide phenotypes, start to do causal inference analysis with potentially modifiable risk factors through methods like Mendelian randomization, and be able to use our GWAS to extract underlying biology of these phenotypes, such as the relevant cell types, biological pathways, drug targets, and the likely causal genes and SNPs. And in the longer term, we're also interested in doing sex-specific analyses, recruitment and sample collection in diverse ancestries through some colleagues in India and Mexico, and combining omics resources for suicide research. So if you would like to get involved in the PGC Suicide Working Group, we're seeking additional cohorts. We've worked with summary stats in the past, but now we can also intake individual level data through the PGC. We need about 200 cases of suicidal ideation or suicide or suicide attempt and 100 of suicide. And that's in order to maximize that trade-off between statistical power and heterogeneity in the meta-analysis. We also have monthly working group calls. We alternate time slots because it's really a worldwide effort, so we try to accommodate different time zones. And I also know there's many people at Sinai who do suicide research, whether it's with omics data, whether it's with clinical trials or clinical risk prediction. So if there's any way that our genetic studies could inform your research or vice versa, we would be glad to collaborate. And so a quick plug, I'm recruiting to my lab at the moment, postdoctoral fellows and students to work with the PGC Suicide Working Group. So please do pass on this information if you know anyone who may be interested. And before I wrap up, I just want to leave you with some final thoughts. Suicide is a highly sensitive topic, and we as a group are very passionate about how we talk about suicide and how we report our research to the press and the public. And the American Foundation for Suicide Prevention have compiled some really useful guidelines on their website about how to report safely and accurately about suicide. So here I've pulled out 3 of their points that I have found most relevant to communicating our research. So to be mindful of the language that we're using to convey the message that suicide is preventable with proper mental health support and treatment, and to provide resources to anyone who may be in need. And I think now that we have this platform as a working group, it is an opportunity for us to set a precedent and draw attention to these issues, so that we can all contribute to reducing the stigma surrounding suicide. And so with that, I will close by thanking everyone involved in this work. It's truly a team effort, and there's 300 scientists here who have worked on these data from all over the world. And I'd also like to thank the participants in these studies, because without them contributing, they're sharing their life experiences and their DNA for research, and this sort of work would not be possible. So thank you again for having me, and I am happy to turn it over to answer any questions if you have them.
All right, great talk. So I'll start us off with a question, and then we've got one in the Q&A, and other people, please post if you have questions. Yeah, my question is kind of vague, but you know, given suicidality is such an environmentally driven phenomena, and the challenges that we know large-scale consortia face when it comes to deep phenotyping. Do you have any thoughts about it moving forward? How do we really go about trying to dissect the genetic versus non-genetic risk factors in a suicide cohort?
Yeah, that's a great point. So it's comparatively easy to combine genetic data across these large-scale cohorts worldwide compared with environmental data. It's more tricky to phenotype the environment and to get those detailed measures and combine them across studies. So here in our Discovery GWAS, we're focusing on looking at lifetime suicide attempt or lifetime suicidal ideation versus control status to get the statistical power necessary to discover the small genetic effects that we anticipate for this kind of complex phenotype. But the great thing about working with such a diverse number and types of cohorts is that many of them have more detailed information available on environmental risk factors and more detailed phenotype data, particularly if they were collected for the study of suicide or suicide research. So this provides us with an opportunity to do targeted follow-up of findings from our main GWAS in some of those cohorts within the consortium, although at smaller sample sizes. So that's why we take this large-scale discovery approach and then do more targeted follow-up in specific cohorts with more detailed information available. Great.
Okay, we have a couple questions here from Stella. So the first one, I'll just read them. The first one is, did you try to identify a relevant cell types from existing omics data?
So yes, we did. We used an enrichment analysis approach through the online FUMA platform. So they have combined single cell expression data from a variety of data sets like PsycEncode, a variety of mouse data sets, and others. And so we didn't see any significant enrichment for any particular brain cell type at the moment. And so I expect that our GWAS summary stats are just underpowered at the moment to do that type of analysis compared with looking at the bulk gene expression in particular tissue types. But it's definitely something that we're highly interested in as the power of our GWAS increases.
And the next question is, death, that says the death by suicide study showed higher SNP heritability versus attempts. Can you comment on this result?
Yes, so I think that this could be for a number of reasons. It could be more homogeneity in the phenotype definition. It could be a more severe phenotype definition. There could be fewer individuals who died by suicide amongst the control group in that GWAS, and all of those could contribute to the higher SNP heritability estimate of suicide. But we really only have that estimate from one study at the moment, so we would need to see that examined in more cohorts before we can really get a handle on the SNP heritability differences between these phenotypes.
All right. I don't see any other questions in the Q&A. So I think we will adjourn. I want to thank Niamh again for terrific talk. Really looking forward to seeing where this work goes in the coming years. And people can get 7 minutes back.
Thank you so much.
All right. Take care.